Hello, and thanks for joining us today for the latest The Scientist webinar. My name is Iris, and I'm here to moderate today's event. I'm very pleased to introduce our speaker, Brian Haldeman, who will be presenting today's talk. Brian is product manager for Lil Tuna at Unchained Labs. Prior to this role, he was a field applications scientist supporting biophysical characterization of proteins and viral vectors. Brian received his PhD in biochemistry from the University of Nevada, Reno, where he studied the mechanics of muscle contraction. His research utilized single molecule microscopy, pre-steady state kinetics, protein affinity and binding characterization, and protein isolation techniques. A very warm welcome to you, Brian. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system to the right side of the live stream player. And with that, I'll now hand over to Brian to begin the talk.
Hello, and thank you so much for the kind introduction. I'm super excited to talk to everybody today about how you can get hooked on automated buffer exchange with Lil Tuna. Now, I don't know about you, but whenever I plan trips to theme parks, I never account for the time I'm going to be spending in line. I'll look at the map, plan out my whole day, then get caught in these long lines that are the bottleneck of the entire day. After the first one, the rest of your schedule is really thrown off. This is how I think of buffer exchange in the lab. It really is the bottleneck of any characterization process, and it's super difficult to plan other experiments around because you never know how long it's going to take. While buffer exchange is consistently the bottleneck of your day in the lab, it's also completely unavoidable. Everyone is doing some sort of buffer exchange. Whether you're doing formulation development on antibodies and need to exchange into multiple different buffers, or designing viral vectors and need to concentrate after purification to get that titer up into a working range, or if you're working with nucleic acids and you need to desalt before sequencing or storage, everyone is doing some sort of buffer exchange. For a long time, these methods were the ones that were available for doing buffer exchange in the lab. And if you're only working with a couple samples, you can get by using these methods. But in a world that's trending more towards plate-based, high-throughput analytics, these methods really can't keep up in that high-throughput environment. So for example, dialysis is a much slower method that often results in diluted samples that require concentration before moving on to any downstream methods. And centrifugation, while much faster, requires you to stand by the centrifuge and requires a lot of manual input. And you also run the risk of clogging your membranes and inducing aggregation in your sample due to the concentration gradients throughout that centrifuge tube. Both of these methods also lack that process control to ensure that all samples are exchanged to the same extent. With TFF, this is a much more complicated process to both set up and also run, and is really more intended for larger scale buffer exchanges. Now, Unchained Labs heard this pain from many different labs and has been developing automated buffer exchange platforms for almost a decade now. And today I'm very excited to introduce to you the latest generation of buffer exchange instrumentation from Unchained Labs, which is Lil Tuna. Lil Tuna is the first fully automated, plate-based, benchtop buffer exchange and concentration system that can run up to 96 samples in a single run. We've really taken the best of buffer exchange and from what we've learned over the last decade and brought it all into one instrument that really is the ultimate solution for all buffer exchange labs. One of the big differentiators for Lil Tuna is that it is compatible with a wide variety of consumable formats. We offer two types of plate-based consumables for high-throughput buffer exchange with different volume ranges, and also UNAs for up to 6 individual buffer exchanges in a single run. This also works great for labs where the throughput needs vary. Some days you may need to do a large formulation screen in an Unfilter 96, and then the next day you could exchange just a few of those top candidates in UNAs. Our consumables come in a wide variety of options, including different molecular weight cutoffs and membrane chemistries. You can find the perfect consumable to fit your molecule as well as the throughput that you require. So let's dive into the underlying technology of how Lil Tuna is able to buffer exchange and concentrate your samples. Unchained Labs developed this pressure-based ultrafiltration and diafiltration method, which provides efficient, gentle exchange with unmatched sample yields. The process starts by loading your samples into the consumable of choice and loading it onto the stage in Lil Tuna. From there, volume measurements are taken for each individual well, and the buffer exchange process begins with applying positive pressure, which forces filtrate through the membrane and retains your sample in the wells. After pressurization, the volume is measured again, and the samples are topped up with the buffer you want to exchange into. This process repeats until your samples have reached their target exchange and is followed by an optional concentration step to lower the volume, effectively concentrating your samples. All pressurization settings are customizable, so if you're working with samples that are prone to aggregation, you can adjust all the settings to keep them well behaved and monodisperse. One part of that buffer exchange cycle I'd like to highlight is the orbital mixing applied during pressurization steps. Without orbital mixing, the sample will build up against the membrane, causing membrane fouling and possibly inducing aggregation of your sample. This frequently happens in centrifuge-based or dead-end ultrafiltration, which necessitates the need to constantly resuspend the samples. Lil Tuna applies orbital mixing to keep samples homogeneous in solution so they don't build up against the membrane. This leads to a more efficient exchange and is also gentle on the samples, which we'll prove in the next few slides. This data highlights both the consistency of high throughput buffer exchanges and the terrific recoveries that Lil Tuna is capable of. Here, we buffer exchanged 10 mg per mil BSA and 96 replicate exchanges to 96%. Final volumes were all consistent, right about 450 microliters, and recoveries were all over 96%. We also used 2 microliters of each sample to verify sample quality by dynamic light scattering, or DLS, on stunner. The consistent sizing pre and post exchange show that the sample quality was not affected by the buffer exchange process. We can also draw the same conclusions when exchanging antibodies in an Unfilter 24. Here, the samples were also exchanged to 96%, but in higher volumes for each sample. This experiment also led to yields above 96% and maintained sample quality that was not significantly affected by the buffer exchange process. Diameters measured by DLS on stunner were not significantly different pre- and post-exchange. One of the most significant developments we made with Lil Tuna is dispensing buffers using peristaltic pumps. This is an absolute game changer when it comes to dispense efficiency because 6 buffers can be dispensed simultaneously into different wells. Gone are the days of patiently waiting for pipette-based dispense systems to slowly dispense buffer into each well. Buffers are stored in bottles off the deck, so your reservoir size is only limited by the size of the bottle. Better yet, pumps maintain the same great dispense accuracy as pipettes, just in a much more efficient format. Here, we're showing dispense accuracy from 25 microliters up to 8 mils across a viscosity range from water up to 50% glycerol. The dispense CV across different volumes and viscosities is less than 1%, showing that pump-based dispense is ideal for high throughput, plate-based efficient buffer exchange. We can also take advantage of the pump-based dispense for concentrating large volumes of dilute sample. Instead of dispensing a buffer used in the pumps, large volumes of dilute samples can be concentrated in the same way. Here, you could fill your buffer reservoir bottle with diluted sample and use that to dispense into your wells. This can effectively concentrate a liter of sample down to 192 mils, which is the maximum capacity of an Unfilter 24. Further concentration is also possible with a subsequent concentration step within the plate. This method is particularly relevant to labs working with AAVs because samples are typically high volume and dilute after purification. This is a great way to bring up the viral titer to ranges that can work for downstream characterization. There are also solutions for samples that are on the other end of the viscosity spectrum. Typically highly concentrated samples that are going through developability or syringeability studies to see how they behave at the concentrations that they will be administered to patients. In this case, we can effectively buffer exchange concentrated or highly viscous sample by switching around a few steps in the buffer exchange process. Before that first pressurization step, exchange buffer is dispensed into the well to ensure viscosity doesn't get too high during the run. Using this method, the sample never becomes more concentrated than when it was initially added to the well, which will lead to the most efficient buffer exchange possible while still maintaining the same great yields and process control. Setting up an experiment in the Lil Tuna software is super simple and can be completed in just a few steps. First, select your experiment type. Then, give your experiment a name and define your consumable type. Next, input the target exchange percentage. type of sample, and concentration range from our predefined, optimized sample protocols. If you want to customize and modify an existing protocol, you can do that here too. Finally, you can paint the plate with both your samples and your buffers. This step can also be done with an imported template, so you can design your experiment in Excel and then upload to Lil Tuna software. Now you just load your plate and place it on the stage, and Lil Tuna takes it from there. During the run, you can keep an eye on progress using the color-coded plate map. Samples in green have reached their target exchange, and the samples in blue still need additional time. The software looks at trends in pressurization times and flow rates to accurately predict how much longer the experiment will take. Finally, on the right side, there is a pause button in yellow, which will display the plate after the current cycle and allow you to remove samples that have completed. This step is totally optional, and alternatively, you can grab a cup of coffee and wait for the rest of your samples to reach their target exchange percentage. When we designed Lil Tuna, we intentionally designed both the hardware and the software to integrate into a fully automated lab environment. With the increased interest in automation and high-throughput plate-based analytics, your buffer exchange can now be integrated into that plate-based workflow. From a hardware perspective, the plates are presented on a sample stage that is easily accessible to plate handling robots. This makes it easy to move your samples downstream into the post-exchange characterization workflow. For software, we have designed a universal API that will allow automation engineers to integrate Lil Tuna into any automated workflow. We also designed the sample import templates to allow for remote experiment design. With these capabilities and the small form factor of Lil Tuna, it integrates seamlessly into Stuntman, which is our fully customizable AI-native work cell that can be designed to automate your entire biologics characterization workflow. Additionally, since Lil Tuna uses plate-based consumables, it integrates really well with other plate-based analytical instruments from Unchained Labs. Speaking of plate-based analytics from Unchained Labs, here's our portfolio of analytical instrumentation that play really well with Lil Tuna. If you want to measure concentration via UV-Vis pre or post-exchange with just 2 microliters of sample, Lunatic is a great option. If you want concentration as well as sizing via dynamic light scattering or DLS, Stunner is in play. If you want comprehensive stability measurements with thermal melting and aggregation, Auntie is the answer. All of these instruments have plate-based consumables, so now you can keep track of your samples throughout the entire buffer exchange and characterization workflow. Lil Tuna truly revolutionizes buffer exchange by bringing together all the right hardware into a compact benchtop instrument. Everything from readily accessible plate-based consumables to efficient and simultaneous dispense of up to 6 buffers using peristaltic pumps. Lil Tuna is suitable for walk-up use or automation labs at any throughput. It really is at home in any lab doing buffer exchange. So in summary, Unchained Labs took our experience over the past decade developing automated buffer exchange platforms and brought together all the best parts into one instrument that fits all sample needs, all throughput needs, whether integrated or standalone. Lil Tuna is the first fully automated, high-throughput, plate-based benchtop buffer exchange system. When it comes to buffer exchange, Lil Tuna is the lightning lane that lets you remove that buffer exchange bottleneck and keeps your entire day in the lab right on track. And with that, I want to thank everybody again for joining the webinar, and I would be happy to address any questions. Thank you.
Thank you very much, Brian, for that wonderful presentation. We really appreciate you sharing your expertise with us today. I'm now going to move on to the Q&A session. To submit a question for our speaker, please enter your question into the box to the side of the live stream player and click send. We'll answer as many questions as possible during the time available. And I can see we already have some questions submitted, so I will move into those. First question is, what makes Little Tuna different from Unagi and Big Tuna?
Yes, I think I can answer that from a couple different perspectives. I think first is the dispense and the way that we do that, right? So Lil Tuna uses pumps, and that allows us to have a much more efficient dispense than pipettes, which were used on both Unagi and Big Tuna. It's also much more automation friendly, right? So it is much more integratable into automation workflows, both from a hardware and a software perspective. And finally, the form factor. It is smaller than both of those systems.
Thank you. And next question is, can the device be integrated with third-party liquid handlers? And are the plates sealed to the external environment? Is the temperature controlled?
Okay, yeah, a few different questions there. Yes, it is compatible with third-party liquid handlers, so TCAN, Hamilton, things like that. Our universal API will allow it to integrate with those systems. Are the plates sealed to the outside environment? No, they're not, because we need to be able to add buffer during every one of those pressure, or after every one of those pressurization steps. So during pressurization, yes, it is sealed from the outside environment while we're applying pressure, but then the plate is presented so that more buffer can be added to them.
And the third question in that series was, is the temperature controlled?
Correct. We don't have temperature control built directly into Lil Tuna, but we do have the option, if you want to, since the buffers are off deck, you can chill those buffers and allow that chilled buffer to be used for buffer exchange.
Thank you. And next question is, why is Lil Tuna considered automation friendly?
Yeah, so I hit on that a little bit before, but I think from a hardware or a software perspective, you can answer that question. So from a hardware perspective, the plates themselves are presented on that sample stage, which allow them to be easily accessed by plate handling robots so they can move on to the next step in the workflow. From a software perspective, we designed both a universal API, allowing it to integrate into automation workflows and work with either third-party liquid handlers or any other automation equipment. And also, from a software perspective, we also have the importable sample templates. So that allows you to design an experiment remotely and then upload that to Lil Tuna when designing that experiment.
Thank you. And our next question is, what is the average pressure applied, and would this be a positive pressure?
Yes, it is a positive pressure, and depending on your sample type and the concentration, we can range the pressure from 15 psi up to 60 psi, so 15 to 60. That is based off of, we have some established protocols that, choose anywhere in that range. But if you want to customize it, you can also choose, any other number in that range as well. So anywhere between 15 and 60 psi.
And how does the system calculate the exchange percentage?
Exchange percentage is based off of your initial volume and how much volume is added to the samples. So it's a volume-based calculation.
Thank you. And how does Lil Tuna handle samples with different flow rates in the same run?
Yeah, so since we are able to measure the volume of each individual well separately, we can calculate flow rates for each one of our samples. So one of the benefits of that is we can work with samples that have differing flow rates from each other. And because of that, we can adjust our pressurization time and really optimize that to make sure that our fastest flowing samples are not drying out, while our slower flowing samples are still, functionally buffer exchanging. So we can really optimize to those fastest flowing samples to ensure they don't dry out, but we are making sure that all of our samples are exchanging in that same process. So, best practice is to have samples that have similar flow rates to each other, but we can compensate for and optimize for experiments that have samples with different flow rates in them.
Thank you. And next question is, what is the maximum viscosity for buffer exchange on Lil Tuna?
Yeah, so the maximum sample concentration, for example, for an IgG, we've seen up to 200 mg per mil that people use, which roughly correlates to about 50 centipoise. So I would say that would be the functional upper limit of viscosity that you would want to use, because anywhere above that, you're just not going to be buffer exchanging as efficiently. So I mean, you could go higher than that. It will just take much longer to do so.
Thank you. And we have time for one more question. Do all of the samples need to have the same initial volume?
Short answer, no. The samples do not have to have the same initial volume. Once again, best practice, we recommend that they're similar to each other, just so your pressurization settings and all that can be best for the samples that are all in your run. But another consideration there is at the end of the run, you have the ability to either normalize by volume, so you can make sure that your samples do end at the same volume, or you can have them end at the same concentration as well. So that's another functionality that we built in here
to make it easier primarily for those that are doing automated workflows to know that your samples are either the same concentration or same volume at the end of buffer exchange.
Thank you very much, Brian, again for joining us today. Fortunately, that's all the time we have available, but any questions we didn't manage to get to will be answered offline as soon as possible. The webinar will now be made available on demand, and you can access the recording using the same link you used today. We'll make sure that any questions submitted on demand are passed along. Certificate of attendance will also be sent by email to everyone who attended today's session. That just leaves me again to say thank you to our speaker, Brian Haldeman, and to everyone in the audience. See you again soon.